And we're going to start today with a session entitled Therapeutic Genomics, where I'm delighted to be joined by some wonderful friends and colleagues who are going to give a series of talks thinking about this phrase and idea of therapeutic genomics. And it's my pleasure to get to introduce this subject to you, the audience. I will start off just to note my disclosures that I receive research funding from BioMarin Pharmaceuticals and also from these bodies here. We heard yesterday about the challenge in primary immunodeficiencies. How do you go from one variant to thinking about 505 genes? We've heard the same thing about thinking about brain disorders, about Batten's disease from Julia. When we start putting this all together, there are 4,500 or so recognized single gene rare genetic disorders. Together, they affect a vast number of people, individually rare, collectively common, a pandemic of rare disease. Millions of people are affected by these worldwide, often in incredibly debilitating, sometimes fatal ways. In the last few years, we've had these game-changing technologies achieve some spectacular successes, and we heard some of these yesterday. The challenge now is, how do we bring those to all of the people who need them as fast as possible? These therapies have some advantages which makes this seem more feasible than that sounds at first glance. If we take an antisense oligonucleotide or a CRISPR gene editing, we can use that genome targeting to target almost anywhere in the genome. Now, that doesn't mean we can currently treat every single variant or every single disorder, but it's possible to imagine with a few technological advances that being on the cards. Because they use genetic hybridization, they bring a level of specificity which is unrivaled, potentially targeting just a single location in the genome with accuracy. Not to trivialize the challenges of off-target effects, but the potential there is huge. Perhaps, though, the most exciting bit is this idea of being able to reprogram them. And Theodore introduced this concept beautifully yesterday of platformization. If you can succeed once in one tissue, in one disorder, in one variant, success in the next disorder in that same tissue is much, much, much easier. Potentially, you just need to change the sequence of that targeting sequence. We've seen examples of this happening in the real world, getting to clinic and making a difference. We've seen it in antisense oligonucleotides, and Tim Yu later is going to talk to us about spinal muscular atrophy and Batten's disease, I presume, and we heard from Julia yesterday. Theodore introduced this example from Intellia of how transthyretin amyloidosis as a CRISPR gene edit in the liver was rapidly reprogrammed to a successful therapy in hereditary angioedema. So if we want to bring these to 4,500 disorders, our challenge is to harness this potential for reprogramming and platformization. To do that really comes down to being a data challenge. What if we could predict that perfect targeting sequence for every single disease-causing variant before we even start going into the design? So let's just imagine what that would look like in terms of real-world effect. So here's an example of a current clinical trial. We identify a target, screen for effective therapies, then dosage and safety towards a clinical trial. Along that process, there are many, many, many times to fail. I've given here an example of 5% success. That's wildly ambitious. That would be a very, very good number to get. But the impact of that low success rate is the cost of it. You're essentially paying for all of the failures along the way. And therefore, that limits, when we think about rare disease, where can you possibly have commercial success? We also saw yesterday all of the biotechs laying people off, the loss of these therapies. In many ways, this is an economics problem. Imagine we could predict therapies before we even start this process with an idea about what might be safe, what might be effective. Even if that just increased the rate of success from 5% to 10%, that would mean you could then move that bar for commercial success to the left. If you could predict it better to 20% success rate, a whole new number of rare diseases start to become viable. And of course, this is an exponential curve. Most of the rare diseases are on the left of this curve. We saw yesterday this lovely description of jammy dodgers, that the idea that if you are just changing the filling, why do you need to go through the entire regulatory process? If you know that strawberry tastes good, it's a pretty good bet that raspberry is going to taste good too. To put this in the context of a therapeutic genomics idea, what if we could predict the next filling? Blueberry, we predict, might be quite good. Marmite. So, how do we predict safe and effective therapies? We're going to go on a bit of a tangent here. So, I started off working in the field of autism genetics. We have known since 1977 that autism is a genetic disorder. This is a groundbreaking study by Fulstein and Rutter. On the left are identical twins. If one identical twin has autism, almost certainly, 92%, the other identical twin has autism. On the right are non-identical twins. If one of them has it, 1 out of the 10 has autism. This then can be used to infer an estimate of heritability. That estimate was in excess of 90%. It is extraordinary that this was known in 1977, given all that happened, for example, with vaccines in the autism field. That study has been replicated again and again and again. Autism, it turns out, is in fact the most genetic complex disorder. This is an analysis done with insurance records at Vanderbilt. So, given that it's a genetic disorder, if you want to understand the biology, genetics is a great starting point. So that launched a quest to find the genes that underlie autism as a route into biology. But for decades and decades and decades, that quest failed. Linkage failed to identify them. We saw an era of candidate gene studies, of genome-wide association. All of it just hit this black box of autism without success. There were a few partial successes of rare syndromes being identified as being enriched for autism, but nothing sort of moving into that main area. And the thing which made the difference was in many ways just technology. But we're going to see it wasn't just the technology so much as the philosophy and the work that went with it. And so that technology was genomic sequencing. Along with that, there were cohorts. For example, here, families where there was one affected child, but also an unaffected sibling control who acted as the perfect control, having grown up in the same environment. From this, sequencing was done, variants were identified, and this family structure allowed the identification of germline de novo mutations, variants not seen in either parent, newly arising in the child. More of those were seen in the cases than the controls, implying a causal relationship of some of these. The question then became which germline de novo mutations actually contributed versus which ones were just passengers. If you line up the mutations seen in the controls, you see them randomly dispersed across genes in the genome. Those are the blue dots there. Everyone has germline de novo mutations. If you take the controls, some of those are also randomly dispersed across the genome, but some of them stack up in genes. So, for example, there are multiple de novo germline mutations in this gene SCN2A. This then became a statistics problem. How many dots in that gene do you need to be able to say that that gene is associated and therefore has a causal relationship with the symptoms? That sparked a change in the way science was done. The ability for this sequencing, but critically the ability to integrate data sets from across the world into one place, meant that people started working together at scale. And it was that working together which gave the cohorts of tens of thousands, which led to success in identifying genes. These numbers here are the number of genes at genome-wide significance associated with that disorder. And this didn't just happen in autism, it happened in developmental delay, it happened in schizophrenia, in epilepsy, in congenital disorders. And we saw yesterday, of course, in primary immunodeficiency. And so now, when we think about the genetic space in developmental delay, these are the genes that we need to target. These are the people being diagnosed every single day in Genomics England. And these are ordered by the frequency with which those are being identified. Collectively, these genes alone probably account for about 0.5% of birth having a severe developmental disorder. So why did this work? Why did we go down this tangent? Genomics was the key to this success. After decades of failure, it was this which made the difference. There were several features of this which, in retrospect, were so important. The first one on the top left is that it was multiple parallel hypotheses. Sometimes genomics is described as being unbiased or hypothesis-free. I would argue against both of those. Nothing is unbiased. And it's not that you're free of hypotheses, but every single gene is a hypothesis. That then necessitates this bottom right corner here. Once you recognize that you're looking genome-wide, you need to correct for that. And it brought a level of statistical rigor, which meant that not only were the findings then validated as they came along, but also prioritized so I could line up the genes as we just saw. Critically, it involved large-scale collaboration. It was as much about the people, the scientists, choosing to share the data as it was about the data itself. And that data had to be generated in a systematic manner so that it was easy to integrate it across the different places. And I think behind this, there was also a case where it didn't matter what disorder you were in, the data was useful. So in autism, we learned as much from samples being collected in cardiovascular or respiratory disorders because they informed our baseline. So suddenly, what was going on across the entire field of all the disorders became relevant to our disorder. And this analogy is where I think we are in that need to get to therapeutics. All of these components can be translated into our area here. So if we think about this transformation, the thing which happened in genomics is we went from candidate genes, small numbers of cohorts, small sizes of cohorts, individual genes. You had to be right if you were going to be successful, and people often weren't. On the right, we saw this expand to large cohorts, multiple genes being done in parallel. And this happened across many, many disorders. We went from a concept of an immune disorder, blindness, developmental delay. Those, of course, still exist, but now they have variants and names and genes. We're now in the middle of another revolution taking place of functional genomics. Here, we've gone from doing, say, qPCR in a single gene in a single tissue to the ability to look with genomic sequencing across multiple tissues and multiple genes in parallel. And in the same way, this data naturally integrates together. So this is an analysis looking at an integrated set of multiple single-cell genomic sequencing from the human brain, and then asking the question, which cell types are enriched for autism? And here we can see on the top line, the answer is excitatory neurons and inhibitory neurons. Of course, critical information when you start thinking about therapies. And so that then brings us to this idea of therapeutic genomics. Can we take those same ideas and apply them here to have the same success? Instead of thinking about one therapy in one disorder at a time, can we start as a community to think about what would it look like to design multiple therapies in parallel together? So how would we apply that? Well, of course, we start off with an absolutely amazing starting point, which we heard about yesterday at the Genomics England and the NHS Genomic Medicine Service. The UK leads the world in the identification, the genetic identification of patients. And so as a starting point to think about this in a genomics way, we can go and proactively search that data to identify the patients who are treatable today. We can start thinking about in silico prediction of therapeutic targets. This is in its infancy right now, but already we can start to make some predictions. Here's an example of a method called Splice AI. You can see here there's an exon with splice sites at either side. In the middle is the motif, so the canonical splice sites of A, G, and GT. You can see there's a wider motif of information. This is how splicing takes place in the cell. Splice AI was trained on the genomic sequence of known splice sites, and then you can see how it recapitulates those motifs in the information of the Splice AI score. What does this mean in practical terms? Well, it means you can search the Genomics England data to identify cryptic splice sites which today are treatable by antisense oligonucleotides. So this idea that you can start to predict what could be treatable from data alone, already starting to work as a concept, but the more data we get, we'll see exponential improvement in this. That can then be complemented by in vitro genomic screens. So this is an example of work done by Nadav Aitov, Jay Shenduri, and myself over in UCSF. What you're seeing here is the gene TCF4. It's a negative strand, so from right to left, multiple different transcription start sites. We're trying to work out how can you upregulate this using a CRISPR activation strategy. So you use a dead Cas9 which doesn't cut, linked to an activating marker, which then can upregulate the gene and potentially as a treatment for the loss of function in TCF4. At the top, every single dot represents a tested guide in a large single-cell sequencing experiment. So all of these dots were generated in a single experiment. And the higher that dot is the stronger the statistical evidence that that leads to upregulation of the gene. So in this one experiment here, you can see that top dot up there in blue. That is the therapy for upregulating TCF4. Now, what was exciting about this was we didn't do one gene. This was a pilot study with 9, but we've just finished doing a study with 300 genes in parallel, designing guides for every single one of those to upregulate them. If we can apply this idea of genomic screening across multiple different ones, we could do this with CRISPR editing, we could do this with antisense oligonucleotides, and we can massively stack the deck in our favor. And then as we move into the piece of taking those and actually testing the actual CRISPR, the actual ASO, the other therapies in tissues, we need to think about standardizing that so we inherently generate data which is ready to go into the informatic piece, creating this virtuous circle. As a key starting point to this, a group of us have started thinking about standardizing protocols for doing this across our sites so we can start to build that information base. Because as this grows, the predictions get better and the entire thing accelerates. We're starting here, but this is, of course, done in an open way, so that we hope it will be a global initiative. We're starting off with 2 disorders which are ready to go, and you're going to hear more about this from James and Yasmina in a minute, of inborn errors of immunity, which of course Theodore talked about yesterday, and retinal blindness. These have the advantage of being treatable with genomic editing right now, with ex vivo editing in the blood, and the retina due to its small size and accessibility. In the brain, we're not quite ready for CRISPR editing, but antisense oligonucleotides are working well. And as we've seen, there are hundreds and hundreds of neurodevelopmental disorders, all of which desperately need therapies. The hope is, with time, to be able to improve the delivery to the brain such that you can then start to bring the genome editing. And this is where I think it gets particularly exciting, and you can start to see how it all becomes a big data problem. That genome editing could target exactly the same site that the ASO has targeted. So the ASO is de-risking your future genome edit by targeting the same symptoms, the same disorder, at the same location, so that once you know that is safe, you can then bring the CRISPR to make that permanent. And then the delivery itself is also essentially a genomics problem. By applying the methods of directed evolution and machine learning, again, there's a really virtuous circle here of data versus wet lab work, trying to bring this all together. And so if we put this all together, we imagine a world where we have changed the way it is done. So this is an example of current development. We have a concept of how to treat a disorder. That then goes into the closed circle, the closed fortress of data-protected, behind closed doors, development of a small molecule. Essentially, it might start off in an open place in academia, but once you've got a lead compound that will go into pharma, you'll hear nothing about it until eventually there's a press release talking about success. That will lead to one therapy. We could do exactly the same with genome-targeted therapies, and to some extent, that's what's happening right now. But if we could open this closed fortress, if we could make all of this data accessible, and all of those patients who are undergoing these risky therapies, having that safety data being made public to bring benefit, we could create the garden, where everything is open, where there's big information commons, so that each therapy can inspire the next one, and that one concept could lead to multiple reprogrammed therapies. But without that data sharing, reprogramming will not work, or at least it'll be one at a time and it will go incredibly slowly. So are we ready for this? I see, with a few advances here, very rapid exponential growth in rare disease clinical trials. We've seen this happen in the diagnosis of rare genetic disorders. 10 years ago, sequencing an individual's genome was incredibly difficult. 20 years ago, it was science fiction. Now it happens daily in a very short space of time, leading sometimes to diagnosis within a few days after birth. The same is happening in functional genomics. It seems likely the same is going to happen in therapies. Are we ready to use our amazing genomic resources to identify those patients at scale for hundreds of clinical trials happening in parallel? Have we supported the patient advocacy groups to do their natural history studies, to organize, to get ready to support those patients through this journey? Have we educated them about this process? Do we have the manufacturing capacity to bring this to reality? Instead of developing one therapy at scale, we're potentially developing hundreds of therapies at small scale, which is a different process to what's gone before it. Do we have the clinical trial unit capacity to deliver these? Antisense oligonucleotides in the brain require an intrathecal injection. It's been estimated that if you treated everyone with Angelman and everyone with spinal muscular atrophy, you would overwhelm this infrastructure right now. And so we need to start thinking strategically. We can see this change coming. How do we get all of this ready to go at the same time? But if we could imagine what the world would be like, a newborn receives whole genome sequencing at birth, already happening in the UK. Through these methods, through this knowledge base we've created, that leads to automated diagnosis and automated design of a therapy. So you don't just get a report of a disease, you get a report of how to treat it. That might go through an automated manufacturing process. Maybe the vector, the Cas9 has already been created. You just simply need to add the guide to it. There's proportional testing of that. There's urgency. These children are sick. We need to get it there, as we heard yesterday. The testing of that is proportional to ensure safety, but in an appropriate way. This then gets sent, and so that when that patient and the family receives that diagnosis, in the next room is the infusion ready to go to treat it so that they didn't even ever get the symptoms they got there. Now, is this imaginable? Well, of course, mailing something in 24 hours remains a challenge, but everything else, we can see a path towards getting there. And so that's what we envisage with this idea of therapeutic genomics. It is about applying the technologies we have, but probably most importantly, it's about philosophy. It is about working together as a community across the world to bring this information to patients. And I'd like to give a huge thank you to all of the wonderful people who started this vision here at Oxford and at UCL, at Newcastle, at the IGI in California. And I will leave you with some work which I did alongside John Lennon and ChatGPT, inspired by Theodore yesterday, of imagining what this would bring. Thank you very much.